like every other person know, those are the common types of genotype. That is, we talk about AE, we talk about AS, we talk about SS, sometimes we talk about SC, sometimes we talk about AC as well too. So these can occur, in variable combinations, so to speak. Depending on the type of hemoglobin, that the individual has. Actually, maybe the normal adult hemoglobin A, or hemoglobin C, hemoglobin S. There are other forms of hemoglobin as well too. That is, we spoke about it the last time as well too. Hemoglobin D, hemoglobin G. There's hemoglobin E, O. So if peradventure these individuals also have or inherit these other forms of hemoglobin, so to speak, alongside maybe a normal adult hemoglobin, then you can have something like hemoglobin AD, hemoglobin AE, hemoglobin AO. Or if they inherit these other forms of hemoglobin with the hemoglobin S, then you can have hemoglobin SD, SG, SO, SE, and stuff like that. So those are also other forms of genotypes that are probably maybe on the African continent or in our own context a little less common, but definitely the common types are the ones that I mentioned earlier: hemoglobin AE, hemoglobin AS, AC, particularly in the southwest, SS and SC as well too. Those are the more important ones, that are probably maybe have of clinical importance, yes, in our own setting at the moment. For example, in AC is like a carrier state actually. Since the person has rather one normal adult hemoglobin, that's the hemoglobin A, so it tends to downplay the effect of the hemoglobin C. But should the individual, have an inheritance of the hemoglobin C and hemoglobin S at the same time, that's the genotype SC. SC also behaves like SS, although they might not have frequent crises like SS, but it's been known that they have almost similar clinical complications, so to speak, as well. In fact, some even say that SC have more clinical complications than SS, with respect to some eye problems and some kidney problems as well too. So they have similar way of presentation as well too. Less commonly, CC can also occur as well too. That is, if you have two couples now that are probably maybe hemoglobin AC and AC, your genotype AC and AC rather, they can have each other as a CC, but in that instance, CC usually will not, have a similar clinical presentation like that of sickle cell anemia, that is SS or SC. So usually, it might not. It's not something that is very, very common like that. So even in my years of practice, I have not come across an individual with genotype CC, but definitely I've seen genotype SC and genotype AC as well too. Not prevalent in the northern part of the country, but usually in the southern part of Nigeria, particularly southwest, where you have hemoglobin C, having some degree of prevalence as well too in that context, so to speak. Someone that is SC is also, let me just use that terminology, in the same shoe as someone that is SS as well too. So there's a limit to which the person can harvest, or let me say fish, for maybe a partner, so to speak. The person definitely has to go for someone that is hemoglobin AA as well too, just like SS, so that if by chance they want to have children, then they can have kids that will have carrier states, that is, have hemoglobin A and hemoglobin S and hemoglobin A and hemoglobin C at the same time as well too. But someone that is SC definitely cannot marry someone. Ideally, well, we are just advising now, we definitely not support it to go for someone that is AS, because at the same time, if you look at the Mendelian pattern of inheritance that we spoke about in the previous series as well too, there's every tendency that they might come down with the child that will either be SS, because there's S in the mix-up, and also C as well too, that is SC. So they can either be SS or SC in the offspring that these couples might have. So it won't be wise, so to speak, to say someone that is SC, should marry or have a life partner that is AS or AC at the same time. The 1 out of 4 was actually like is a misinterpretation. Let me just put it that way. I would not want to. I would not want to say I flog the biology teacher, so to speak. You understand? I think it's understanding now. We're in the jet age now, and understanding is getting clearer by the day. So the risk is actually in each pregnancy. I have to re-emphasize it again. It's in each pregnancy, not that when a couple there's AS and AS marries, and then maybe it's the last child that will be SS. No. It's in each pregnancy that there's a 25% risk that the offspring or the child that they are about to give birth to will have SS. So we don't know the position that it will be, whether it's the first pregnancy, whether it's the 10th pregnancy, whether it's the 100th pregnancy, so to speak. But in each pregnancy, there's every risk that 25% of the outcome of that pregnancy will turn out to be SS or SC, depending on the variant hemoglobin that the couple have at that point in time. So it's not 1 out of 4. It's actually like a ratio, it's like 1 in 4. So 1 in 4 should be the 25% that we're talking about in each pregnancy. So it's actually a misinterpretation. And now we are out to correct that misinformation by saying that the risk is in each pregnancy, not that, okay, when you have your first pregnancy to be EE, the second child will be AS, the third child will be AS, and then the fourth child is destined to be SS, so you will not have it. No, it's a misrepresentation, it's a misinformation, and that's why we are out here to correct it. It's in each pregnancy. Well, in the actual sense, genotype will not flip, because it is your genetic composition. So it's like saying that your genetic element in your body flips or changes, so to speak. It won't actually, except if the person has undergone a form of a transplantation that is changing or altering the genetic composition of that individual. So once you are one particular genotype, it does not change. It can't change. I guess what happens is that there are some individuals that you say, yes, they are SS, and they don't experience any clinical symptoms. Yes, it can happen. Probably maybe some of those individuals have what we refer to as some disease-modifying genetic factors, so to speak. Maybe some of them have persistent fetal hemoglobin in their system. Some of them have coexisting beta thalassemia or alpha thalassemia that might modify the severity of the disease, such that they don't present to the hospital as frequently, as you expect them to be. So that one too can happen. But genotype flipping does not happen, except in the context of what I just explained earlier, somebody going through transplantation or altering or changing his genetic composition. So genotypes usually will not flip. There are cases in which people have children and then they are always sick, you understand, and they say, this one is a sickler. No, sometimes it does not happen that way. I think I've heard of that scenario as well too, in which they say the child is AE, but it's always behaving like a sickler and things like that. Children, manifest in different ways, particularly when it comes to maybe handling infections, maybe when they are still in their growing period. So it is not because someone is always sick that labels the person a sickler. Sorry, I'm using that terminology, sickler. I'm not actually supposed to use it, but so that at least you can understand what I'm trying to say. So it's not that someone is always sick that labels the person as as a sickle cell disease person or patient, so to speak, or someone living with sickle cell anemia. No, it's until you have your hemoglobin electrophoresis or hemoglobin phenotype done as a person living with sickle cell anemia. So in actual sense, genotype or hemoglobin phenotype will ideally not flip, except in the context of a stem cell transplantation. Now, from the word genotype. Genotype means gene typing. That is the genetic composition or constituent of an individual. When it's being tested, it's what is referred to as genotype. You're actually looking at the genes that are responsible for the production of a particular hemoglobin an individual has. For example, now, this gene testing, it's on a genetic event. It's a genetic event on a genetic level. There are what we call chromosomes. I'm sure everybody knows the context of chromosomes. Chromosomes, alleles, DNA, you understand, that occur, or let me say alleles and DNA, that occurs in the chromosomes of every individual. Now, the chromosome that is responsible for the production of the hemoglobin chain, there are actually two. There's chromosome 16, which codes for the production of the alpha light chains. We call them alpha light chains. And chromosome 11, which codes for the production of the beta light chains. Now, I'm breaking it down now. hemoglobin contains of 4 hemoglobin chains, 4 chains. There are 2 alpha chains and there are 2 beta chains. Now, the alpha chains is produced by chromosome 16 and the beta chains are produced by chromosome 11. Now, on those genes, so to speak, there are what we call codons. Now, these codons are responsible for protein production and amino acid synthesis in the body. So, for hemoglobin, on the chromosome, on the sixth position of the beta chromosome, that is chromosome 11 now, or the beta genes on chromosome 11 on the 6th position, you have GAG. Normally, there's a codon called GAG, which produces glutamic acid on that amino acid chain sequence. There are actually 141 of them for the beta chain. So on that position 6, GAG is changed to GTG. Now, the resultant effect of that change is that GTG will now produce what we call valine instead of glutamic acid. So that replacement of glutamic acid by valine is what makes the hemoglobin of the sickle cell individual sickle. Anytime he's exposed to any form of stress, whether infection, whether any form of emotional trauma or whatever, the hemoglobin will sickle and then they have those kind of symptoms. So that's on a genetic event. So when you go to the genes, to the DNA on the chromosomes now, and you try to identify this codon, that is the GAG, GTG, UGG, whatever it is, that is what is called genotype. That is, you are typing the gene that is responsible for the production of this particular hemoglobin S. That's genotype. Now, the phenotype is the physical expression of the genetic constituent of an individual. I'll break it down again. The reason why we are black is because we have a gene that is producing melanin, that is making us black. The reason why our eyes are brown is because we have genes that is making the color of our eyes brown. The whites, they will be either blue or green or whatever, you understand? There's a gene that codes for all these physical expression of ours. The reason why your hair is black, there's a gene that is responsible for it. So the outward appearance of the effect of a gene is what is referred to as the phenotype. That is, the physical appearance now is the phenotype. So now, if you subject this hemoglobin to electrophoresis, either in acidic medium or in alkaline medium, they tend to move at various speeds, and that's how we come about the principle of electrophoresis. That is, whether someone is AA or someone is AS or someone is SS, is depending on how this hemoglobin move on that paper. So because you are seeing it move, that is the physical expression now of the effect of those genes now that we are seeing now when we subject hemoglobin to electrophoresis. So that's why we say it's hemoglobin phenotype, not genotype. And like I said, there are different types. In fact, there are like almost over 200 of almost 400 types of hemoglobin that we can talk about, you understand? And it is a physical expression of genes in our human body that produces these types of hemoglobin. So that's why genotype is different from phenotype. Phenotype is the physical appearance, that is the outward appearance, what the eye can see of an individual of a particular protein, while the genotype is the genetic composition, so to speak, that is responsible for that physical appearance. So there are two different things entirely. I hope I have not confused our listeners by talking about genotype and phenotype on this level. It's purely scientific work, and I'm sure students of science will understand everything that I've just spoken about right now. There are numerous other types of tests that can clearly identify the type of hemoglobin you have, not until you do the genotype. So like I said earlier, the hemoglobin electrophoresis is also one of the good tests as well that they can use. There are other ones, isoelectric focusing, you can do capillary electrophoresis, but there's a gold standard, so to speak, which we call the high performance liquid chromatography. That does not just identify the hemoglobin, it also quantifies it, so that you know that, okay, the amount of hemoglobin you have, whether it's in the right range or whether it's not in the right range. So we know that, okay, there's probably a problem with this individual. and then we can subject that person to further testing to know what the problem is. So you might not need to do a genotype, but a phenotype, yes, and probably maybe an HPLC will suffice, in that context. Well, in actual sense, anyone that has the anemia or that's suffering from anemia, the body has a physical way of adaptation, so to speak. It's like an adaptation of adapting to the anemia, such that the intestines increase iron absorption from diet. Usually you get iron from diet. Either you eat meat, either you eat fish, either you eat green leafy vegetables, or some individuals that actually drink blood or cook blood, something like that, like the Maasai in Central African Republic or Kenya. So you actually get iron from such sources. So anywhere that has anemia tends to upregulate, iron intake and iron absorption. Is a physical, it's an adaptation, But in sickle cell anemia, it depends. Usually, iron is a tightly regulated element in the body, in the sense that once you ingest it or once you eat it, there is basically no way that you can excrete it except you bleed, so to speak. That's the only way you can lose iron, or except you have a condition that's making you bleed, like ulcer, or like in females now that have the monthly menstrual cycle, or some of them that have what's it called again, uterine fibroids, and then they bleed excessively, then they tend to lose iron in such instances. But usually iron is tightly regulated. Once it's inside the body, it does not go out. So that is one of the reasons why for sickle cell patients, because their body is breaking down the hemoglobin, you understand, the iron is actually not lost, it's actually being recycled in the system. So you adding iron to them in such instance might make them overload, so to speak, have excess iron in the body. That's what I mean by overload. So in that context, it might not necessarily work out for them. But we also know that for females that have sickle cell anemia and they lose blood, after a while they tend to go into the negative iron balance. So some authority will actually suggest that for females, you might actually place them on iron supplementation because they are losing blood every month. You understand? And if you don't try to replace that, it might worsen the anemia they have. Another set of scholars, we believe that sickle cell anemia is a chronic disorder. Inflammation, hemolysis, everything going at the same time. And in the body, when there's inflammation, inflammation tends to downplay iron uptake and usage. So after a while, they may tend to have some form of iron deficiency. But it's only when you test it and you see that, okay, yes, they have iron deficiency, then you can give them iron supplementation. That is after testing in the laboratory. But to just give them iron supplement just like that because they have sickle cell anemia, it's not something that we practice routinely. We don't practice it routinely, except we have evidence to show that, yes, they are actually losing iron. Ideally, they maintain their iron status. They don't tend to lose it, you understand? But if there's any other clinical condition that they have on top of the sickle cell anemia that is not making them use iron, then you might need to give them the supplement, so to speak. And again, some of them actually are on blood transfusion programs, like particularly in children and young adults that probably maybe have had stroke as a result of the sickle cell anemia, and then they have to take iron transfusion almost every month. So in such cases, they may tend to have excess iron as well too. They may tend to overload. But there are also preventive measures that we will set in place to prevent them from having iron overload as well too, by chelating excess iron in the body, giving them some drugs or some infusions, such that it doesn't cause another untoward side effects apart from the complications of the sickle cell anemia that they are already dealing with. So routinely, we do not give them iron supplementation. Routinely, we don't.